It's my big pleasure to have a visitor talk today with two visitors, good friends of mine, that I know one of them, I know him from a long time now, and it's Jamie Neer. So Jamie is coming from Toronto. He's working on, so he will be the first speaker, and he's working on MR spectroscopy since a long time. And I remember your talk at ISMRM. So every time that there is an ISMRM educational on spectroscopy, you are the one who's more often invited to talk about quantification, to talk about fitting. When we do workshops, you are there to talk about quantification fittings. Last year, you were talking about organoids. For the first time, it was not quantification, not fittings. So Jamie is working a lot on, as I told you, spectroscopy in animals, in humans, and a lot of processing. The consensus paper on processing on quantification, which was published in 2021, 2022, I think, in Animal Medicine. So Jamie was the principal writer of this paper, or the organizer of the paper. So yeah, I think that this is Jamie. And it's our pleasure to have him around. He is also invited in the PhD defense of Brian, who will take place Friday morning. So he will be cooking for Brian Friday morning. But until then, Jamie, so as I told you, Jamie is working in Toronto now, University of Toronto. He has his own lab. He's also teaching there. And he's a scientist at Sunnybrook Research Institute in Toronto, and of course, associate professor in the Department of Medical Biophysics at the University of Toronto. And Jamie is working since recently on cerebral organoids. If I remember correctly, they are human organoids. Mm-hmm. And it's something that we are also trying to implement here and scan them in vivo in a bioreactor that was built by TAN, which is their TAN. And Eloise is in charge of this project from our side, and she works in collaboration with Pauline and Christiana and other people who are on Zoom. So it was a big, a big thing for us that we have Jamie here, and that we invited him to talk about organoids, about his work on organoids. So he will be explaining us how he's doing high resolution magnetic magic angle spinning, Usher mass, as we call it, NMR spectroscopy in these organoids. The floor is yours, Jamie, and thank you so much that you accepted to come first for the PhD defense and then to talk about this amazing project.
Thank you very much, Christina, for the introduction and for inviting me. Very happy to be here. And this is I'm going to talk about HR mass in cerebral organoids, which is a bit of a new topic for me, and a topic I'm excited about. But what are organoids? Maybe it was about 5 or 6 years ago I heard about organoids for the first time. I've been working in spectroscopy for a while. I've been doing imaging in humans, also in rodents. But then I heard about organoids and I thought, oh, this is really cool. So what are organoids? I think everybody here knows they're self-organizing, three-dimensional cultured human brain tissue, or can be other species as well. And they're derived from stem cells and can be reprogrammed into a neural fate using transcription factors. And these are 100% genetically human. They can be patient-specific, so you can make organoids from patients with different diseases to study the brain tissue that has genetic factors that actually result in disease. They can grow between 2 to 4 millimeters in diameter. They can have electrical activity like a real brain does. I think there's work on having them have vasculature, although they normally don't have vasculature in their normal state. But so I thought this was a really interesting new technology and a great way to study brain tissue at a very close level, the way it is difficult to do with real brains. And being a spectroscopist, when I learned about organoids, I thought, well, wouldn't it be cool to do spectroscopy in these organoids? So what are the big advantages of cerebral organoid research? Well, they allow us to study human brain tissue using invasive techniques that we wouldn't otherwise be able to do. So you look at them with immunohistochemistry, microscopy, electrophysiology. We can do very detailed genetic analysis from up to single cell RNA transcriptomics. We can do pharmacological challenges on them. So there's a lot we can do with organoids that to kind of let us study the brain at a detailed level that we might not otherwise be able to do. And they might, another advantage is that they may recapitulate human brain physiology better than, say, a rodent model does. So we can do a lot of these things in rodent brain, but are they a good model for the human brain? Maybe not. So organoids may provide a bit of a better model. They're not perfect either, obviously, but and then also they're potentially a platform for human disease research. So we can derive organoids from patients with different diseases, like say Alzheimer's disease, ALS, as I'm going to talk a bit about near the end, and maybe individual subject organoids who model a particular subject, and then we could do test different drugs to see if maybe we could get a personalized approach to medicine using this patient-specific organoids. So these are some ideas of why organoids could be useful. I'm sure people have even more ideas than what I just told you. But I was always interested in the fact that if you look across different species, human, rat, and mouse, anatomically, they don't always look very similar, right? You can see that the brain of a human is not very recognizable next to the brain of a rat or a mouse. But if you look at them chemically, they are very similar, right? You have a lot of the same metabolites in rat brain and mouse brain and human brain. The only major difference is maybe the taurine levels are higher in a rat and mouse, but otherwise, they look a lot alike. And so, like I said before, when I heard about organoids, I wondered whether maybe we could do something similar. Could we do spectroscopy and see what the spectra look like in organoids? So hopefully today I'll show you what they do look like. So the objective, I guess what I'm going to talk about today is to develop an NMR spectroscopy technique for assessing neurochemistry in intact human-derived cerebral organoids. And rationales being, so one, do they accurately model human brain-like neurochemistry? If not, why not? And can we improve the way that the organoids are engineered to make them better model human brain chemistry? That obviously I won't tackle this whole question in this talk today, but down the road, hopefully maybe we can address that. Can we understand the cellular, genetic, epigenetic correlates of specific neurochemical markers? And maybe I'll get a little bit into that question in this talk. And then finally, can we identify and understand neurochemical alterations in disease? I won't answer all three of these questions in this talk. I'm really just going to show the methods that we've used to get spectra, because for me, this project is really new and we haven't really delved into these questions of actually applications of it yet, but in the future. Sorry, the name is cut off there, but that's Dr. Carol Sherman's in the top right. And when I first moved to my current hospital, the Sunnybrook Hospital, Sunnybrook Research Institute, I met Dr. Carol Sherman's, who got a lab that develops cerebral organoids. So this is not something I do in my own lab. I couldn't make organoids, I don't think I was asked to. But luckily Carol does, and she's really interested in neurodevelopment. So she studies the developing brain and also studies how cell fates are altered by different transcription factors and things like that. So she makes organoids. And when I spoke to her about the idea of doing spectroscopy in organoids, she was interested in, and that's how this collaboration started 4 or 5 years ago. So this is a description of her method for generating the organoids. And I don't know too much about it, but I think she's using something called the Lancaster method, which is it's a protocol for developing organoids. There's another method called a Pasca method, maybe you're familiar with. While previously she was using the Lancaster method, she's now experimenting with the Pasca method. And all of the organoids I'm going to show you in today's talk were developed using the Lancaster method. So when she first gave us some organoids, this is, does that work?
No, it's a virtual one.
Oh, it's a virtual one.
Yes, you can choose on your computer.
Other pointer, this one. So that's the organoid which we put into a test tube, and we thought, let's try doing spectroscopy on it in our 300 megahertz vertical bore NMR system. So we did this. Oh, I'm sorry, the text actually doesn't show very well here, but We scanned different days of maturity. So we cultured the organoids for 70 days, 119 days. We put it in sort of like a mixture of media and D2O. And we used our standard surface coil. And I'm so sorry, a lot of the text is cut off, but we did some imaging experiments just to actually see if we could image the organoids. And I'll show you the results there. So this is the MRI images acquired, and I've kind of colored in the organoid in blue there. The diameter is about 1.8 millimeters in this particular organoid. And we estimate the volume was about 4 millimeters cubed. Okay. So then we tried to do spectroscopy. We used just a localized spectroscopy sequence. So we put a volume of interest around the organoid. And these are the spectra that we got. So we initially had these kind of not so good looking spectra. Yeah. We couldn't really identify any of the metabolites in those spectra. We didn't know what we were looking at. It didn't look like your typical nice rat brain or human brain spectrum. And so we didn't know what to do. So we did some reading. And what we found out was that, here's some peaks: lactate, lipids, glycine. That's what we think they are, but we're not even really sure. Maybe macromolecules. So after doing some reading, we learned that a lot of people, when they're studying small tissue samples or semi-solid samples, they use high-resolution magic angle spinning. Okay, so this is a technique for chemical analysis in small solid or semi-solid samples. Prior to HR mass, NMR was mostly restricted to liquid samples because solids have a very, very short T2 and with broad line widths and don't give nice spectra. They give mostly uninterpretable spectra. But with HR mass, you can produce this dramatic line narrowing effect, which allows us to see very sharp peaks and make better interpretation of the spectra. So how does it work? Well, to understand how it works, we have to know about the importance of molecular motion and its effect on line width. So maybe most of you know this already, but I'll quickly walk through it. If you just look at a glass of water and you were to do a spectrum on it, you could get a very nice narrow line width of about 0.1 hertz. Okay, so you get the signal persists for a long time in the time domain, and that gives us a sharp peak in the frequency domain. But if you took the same water and you turned it into ice and you took a spectrum, suddenly your time domain signal disappears, it decays extremely quickly, and your spectrum gets very broad. So there is a peak here, but it's got such a broad line width that you can't really see it at all. Okay, so it's just the difference. The difference here is related to the molecular motion. Okay, so it's also related to the motion of molecules within each other's dipole fields. So if you think of each spin, each spin as being a magnetic dipole, it's surrounded by this cloud of magnetic field distribution, where you have a positive magnetic field here and here, and a negative magnetic field here and here. So there's very inhomogeneous magnetic field surrounding each dipole. And the equation of that field is given by that equation in the middle. And so all of the spins that are moving around in the vicinity of this dipolar field, and that causes them to experience changing magnetic fields over time. And if you think about, say, 3 different scenarios where you have 3, on the top we have a solid where there's very little motion. In the middle we have a semi-solid where there's some molecular motion. And on the bottom we have a liquid where there's lots of molecular motion. I'm going to show a little simulation of what the phases of each one of these spins would look like over time. First, I'm going to show how their magnetic field offsets are going to change over time, and then the phase. So here's the first movie. This is a solid. So you can see that these little spins are moving around in the presence of this inhomogeneous dipolar field. And on the top, you can see that they have magnetic field offsets that are fairly continuous over time. The magnetic field that each spin experiences doesn't change very much. And as a result, they're going to dephase consistently. So you can see they're all the phase of each spin is like fanning out. And over time, you get a lot of dephasing. And that means T2 decay, lots of signal decay. Okay, but what happens if now you have more molecular motion? If you have more molecular motion, the field offsets are less constant. So they're moving into different field regions, and the field offsets are changing. And that means that instead of fanning out so much, you get some cancellation of the phase offsets, and you get more phase coherence over time. And it becomes more clear when you look at a situation where there's lots of molecular motion. So here, now they're moving around a lot. The magnetic field experienced by each spin is going up and down and up and down and changing. And that tends to average out to 0 over time. So you get less and less phase dispersion. Now you have more phase coherence. And all of these spins are going to stay coherent, and they're going to have a signal that persists for much longer. Does that make sense? So the idea being that the more molecular motion you have, the longer your phase coherence and your signal will persist. So that's kind of the concept of molecular motion and how that impacts signal relaxation. And as early as 1954, Felix Bloch had suggested that they realized the importance of molecular motion and suggested that if you might be able to simulate molecular motion in solids by spinning the sample, which was a pretty impressive thing to realize at that time, 1954, when NMR was pretty much brand new. And by 1959, the method of spinning the sample had become formalized by these two, Raymond Andrew and Irving Low, and this gave rise to the idea of HR mass, or magic angle spinning. The actual part about the magic angle is related to the angle at which this dipolar field is 0. So if you take that dipolar field equation and you solve for the theta in which the B field is 0, you can see that you get this magic angle theta m, which is the magic angle that's 54.74 degrees. So this is your magnetic field. This is the main magnetic field direction. And at an angle of 54.74 degrees, the magnetic field is 0 everywhere along that axis. And so that's the magic angle. And why is it important? Well, if we rotate the sample about an arbitrary axis, we tend to reorient the dipolar field offsets in the direction perpendicular to the axis of rotation. And this causes the field offsets to average to 0. However, it's not possible to remove the field offsets along the direction of the rotation because you're not changing anything in that direction. So by putting the direction of rotation along the 0 axis, there's no field offsets along that direction. So you get the most benefit of spinning when your axis of rotation is along this magic angle. Okay. All right. So hopefully that clears up magic angle. Maybe you already knew that, but in case you didn't, hopefully that was useful. There's other line broadening effects besides dipolar coupling, and so things like chemical shift anisotropy and macroscopic B0 inhomogeneity, they also contribute to line broadening. And HR mass can also help remove line broadening effects that are related to those issues as well. Okay, so one other side effect of HR mass spinning is that you get something called spinning sidebands. So while rotating spins are traveling through field inhomogeneities in a cyclical or periodic pattern, the Larmor frequency increases and decreases in a periodic way according to this equation. So you have your main magnetic field plus some delta B0 and then cos WRT, which is like omega R, which is your rotational frequency. And you can rewrite that as a sum of frequency components like this, where you have one frequency component at the main Larmor frequency, and then you have copies or like additional components at integer multiples of the rotational frequency. And so you're going to get peaks in your spectrum at these additional frequencies that are related to the rotational frequency. And those are called spinning sidebands, and I'll show you what that means in the next slide. So if I don't spin my sample, I just have a peak in the middle. But if I start spinning my sample, I start to see these copies, these extra peaks, and those are called the spinning sidebands. And the faster I spin my sample, they push out further and further. So I need to spin my sample fast enough so that the first spinning sideband is outside of the spectral range of interest. So that's important to carefully choose your rotation frequency. Okay, so HR mass, how is it performed? You place your sample into a rotor, which is like a little cylinder. You orient the whole system at the magic angle to the B0. So there it's oriented. The rotor is surrounded by a solenoid, which is your RF coil. Your sample ideally is going to be in the middle of the rotor. And then you have two air sources. One is called the lift air, or the called the bearing air, and that kind of levitates the sample. And then you have another air source that spins the sample. That's called the drive air. And it spins it quite quickly. The rotor has a little black mark on it, and with an optical sensor, it can detect the rotation speed, and you can use it to control the rotation speed very carefully. Okay, so these were our first NMR results. We went from our first experiment where we had spectra like this, but with the same organoids, not the exact same organoids, but identically prepared organoids, and we started doing it with spinning, we began to get spectra that look like this. And so now you can see a lot of metabolic details that we wouldn't have been able to see before. So there's no need for tissue extraction. This is an advantage of being able to do HR mass, is that you can do NMR on tissue samples, but you most of the time you have to do a tissue extraction. And sometimes if you do that, there's a risk of losing information or altering the metabolism. So this is a way to avoid possible issues associated with tissue extraction. You might be able to do longitudinal studies with HR mass. You can do studies with pharmacological interventions. So you could do an organoid that was intact, maybe add a pharmacological agent and see what happens after treating the organoid with, say, a drug. And you can also do follow-up measures. So you could, after doing your HR mass, you could do immunohistochemistry or genetic testing to look at other features. So it's kind of non-destructive. I guess that's the main advantage. I'll show a few examples from the literature. Here's some work done by Martin Wilson where they looked at, this is not in organoids, but they used surgically resected pediatric tumor samples and were able to get really beautiful spectra that they could use to better understand the metabolism of these different tumor types. So I won't spend too much time on these examples. Here's another one by Leo Cheng. Leo Cheng is a researcher in Boston who does a lot of HR mass. And he uses, studies brain tumors, malignant lymph nodes, breast cancers, all intact tissues, and typically analyzes them using principal component analysis to look at differences between different tissue types and different cancer types, things like that. This is some work by Andre Simpson, who works in Toronto and is actually a researcher that we were fortunate to collaborate with on this project. And Andre has developed what he calls comprehensive multi-phase NMR. So it's a way of separating the components of the HR mass signal from sort of liquid state versus solid state and gel, like semi-solid state, and get separate spectra for each of them. And he's also done a lot of studies in live organisms to look at the effects of spinning on the organism. So how fast can you spin them while they still maintain like viable, they stay alive basically. And it showed that at low spinning rates, organisms can survive 4 to 6 hours spinning experiments without any, without signs of death, which is impressive. And that's spinning them at like 2,500 hertz, I think. So, okay, so we were fortunate to work with Andre Simpson, and for these first experiments, we used a 500 megahertz Bruker NMR system. We have 12 organoids that we scanned, and they were ranging in age between 85 and 312 days of maturity. And we placed them in a 4 millimeter rotor with an insert to keep them in a small volume within the center of the rotor. We spun them at 2,500 hertz, and we maintained them at 5 degrees Celsius during the experiment. And we used an 11-second TR and 512 repetitions. And so one of the things we also did was it's called inverse diffusion editing. So in the spectrum, there are signals from small molecules like metabolites, but there's also lipids and macromolecule signals that make it a little hard to quantify some of the metabolite signals. So we acquired a sort of a conventional spectrum like this, but we also acquire a spectrum that's heavily diffusion weighted, which tends to delete the metabolite signals since they're very freely diffusing, but it does leave the macromolecule signals, which are more immobile. And then we can do a subtraction to remove the macromolecule signal from this spectrum and get a spectrum that's more pure metabolites. And so that helps for quantification. And here's a closer look at the spectra. We're able to quantify, I think, somewhere between 17, a little over 17 metabolites in the spectrum. And some of them are the ones that we normally observe in the human brain, and some of them are a little different. We had some differences. Oh yeah, here's a look at all 12 of the spectra from the organoids, and they look pretty reproducible, and there's some minor differences between them. And if you look at the concentrations, well, we tried to estimate the concentrations, and we found a few notable differences between organoid spectra and human brain spectra. One is that, first of all, there's a very low NAA concentrations in the organoids. In the spectrum, you would expect NAA to appear around 2 ppm. And if you see, most of the spectra don't have a strong peak at 2 ppm. So there's very little NAA in the organoids. So that's one difference that we noted. We noted this peak here at 2.6 ppm. You can see most of the spectra have a strong peak there. That's hypotaurine. And they don't seem to have a strong peak from taurine. So organoids, interestingly, have high hypotaurine, but not much taurine, which is also different to what we see in the human and rat brain and rodent brain. Lactate, they had very high lactate levels. We expect this is likely due to hypoxia. We're not giving them any oxygen during the experiment, so they're probably getting hypoxic. And then the other thing we noticed is a relatively balanced level of glutamate and glutamine. So we know that in vivo, the brain has a much higher level of glutamate, and like a 5-to-1 glutamate over glutamine ratio. We found that the levels of glutamate and glutamine were pretty equal in the organoids. So we did, I'm not going to talk about it here, but we did some RNA sequencing in the organoids to try and understand some of these differences to, differences to the human brain. But I'm not going to show it in this talk because I didn't want to take too much time. Okay, so we observed some substantial differences between chemistry of organoid tissue and in vivo brain tissue. The main four are the ones I just mentioned. The issue of the low NAA, this is also observed in fetal brain, and we wonder if maybe this is indicative of the fact that organoids are really a model of early brain development. They're not really a model of adult human brain. So, that's something we want to look more into. Maybe if we wondered if we cultured the organoids for longer and longer, maybe they would eventually begin to develop an NAA. We have gone all the way to 312 days and still haven't seen anything, but maybe even further, we could start to see NAA. The quantification is challenging. We tried to measure the quantification using LC model style approaches, but because the peaks are so sharp and because the basis sets that we're using are not 100% accurate, any little offsets cause LC model to basically fail. Whereas in the human brain, the peaks are broader and a little bit of offset won't hurt you that much. But in organoids, if you're off by a little bit in the basis set, it doesn't work so well. So we're still working on that. And another limitation of what we did was that we cultured the organoids at our hospital, but the place where we did the NMR was about like a 20-minute drive away. So we had to transport the organoids. We're wondering about the effect that might have had on the metabolism. For example, the lactate levels probably got higher than they normally would from being out of their culture for a long time. And so in the future, we're hoping to do these experiments right on site. And we now have an HR mass probe, which I'm going to quickly show you, that we got for the site at Sunnybrook. And so this is a picture of the probe. We recently got it in the summer, and we purchased it from Doty Scientific. You may have seen their booth at the ISMRN sometimes. It's a company in the States. So that's the probe. It's tilted, and the air. This is the air that comes in to spin the organoid. This is a picture of the rotor. So the rotor looks like that. And inside it, we have a little insert that keeps the organoid within a certain small volume, so that it's not leaving the sensitive region of the coil. Those are called sealing cells, the inserts. This is the spinning speed controller. So this thing detects the spin rate and then adjusts the air pressure to maintain the spin rate at the desired level. This is also tricky to get right. That's a picture of our NMR, and this is a picture of the probe when it's inserted from the bottom. You can see lots of stuff coming in. You have RF lines coming in and out, but you also have gas lines. So this is a gas line for the bearing pressure, and this red one is a gas line for the rotor pressure. We're also going to have a temperature control in there to keep the, so they're going to be sending in cold air to keep them at like 5 degrees Celsius. So we're trying to figure all this stuff out at the moment. And this thing here is called a bench spinner. Sometimes it's really hard to get the sample to spin properly. It won't spin at the right rate or whatever. So the bench spinner is a way to test that the rotor is balanced properly, and you can test the spinning before you put it into the NMR to make sure it all works. So yeah, now we're still working on getting the temperature control implemented. This is a spectrum from a sucrose solution, one of the first spectra that we've acquired with our new HR mass probe. So not much there. It's a liquid sample, so it wasn't really hard to get a great spectrum, but at least we got our new probe working, and hopefully I'll be soon showing you new spectra from organoids on our new HR mass probe, but I haven't got them for you yet, I'm sorry to say. And eventually we want to use this platform to study genetic risk factors for neurodegenerative diseases. So that's something my lab is interested in, is neurodegeneration like Alzheimer's disease and ALS. We know that each of these diseases has specific genes that are associated with risk. For ALS, there's a gene called SOD1, which is a familial mutation that is associated with early onset ALS. And so we can, with organoids, we can generate organoids that have the SOD1 mutation, and we can also generate isogenic controls that are identically genetically other than the mutation. So we can delete the mutation basically. And then we can see by scanning groups of organoids with and without that mutation, how do they differ metabolically? And the same thing with C9orf72. This is a mutation associated with ALS. We can see what does that mutation do to metabolism. And there are good reasons to believe that those genes will affect metabolism. And maybe they can help us identify neurochemical biomarkers of those particular diseases. Likewise, with Alzheimer's disease, we know that ApoE is a genetic risk factor for Alzheimer's. It's related to lipid metabolism and lipid transport. What do the spectra look like when we specifically manipulate that gene and to look at the different alleles, the different risk alleles of ApoE? We hope to be able to do that with our organoids. Same thing with PS1, presenilin-1 mutations, another risk gene for Alzheimer's. And so this is just a few of the experiments that we're planning. I'm sure there's many more that we could plan, but I think first we have to show that we're actually able to detect signal changes in organoids, and that hopefully will come soon. So hopefully I've shown you that HR mass is a valuable technique that simulates molecular motion by spinning the sample rapidly. It's a powerful tool for metabolic analysis in cerebral organoids, and it may provide insights related to genetic risk factors in neurodegenerative diseases. And thank you to everyone I worked with. This is circling the main people who were involved in this project. And special thank you to Christina for inviting me here. And I didn't take any questions.